happy to be here to give a talk today, and I'm going to take a little bit of a different tack from the last talk, which I thought was fascinating. I know one thing for sure. I'm not sure whether I want to be buried with my cell phone or not, but I definitely want no access to email because I don't need any more of that, but I'll think about the phone. But our approach is a little bit different, maybe even, I don't think it's uncomplimentary. I just think it's a different tact on aging. And what we're really trying to do is to look for strategies in the biomedical sector to slow down the aging process, but not so much to keep people alive longer, although that may happen. Our main goal is to keep people healthy longer. So when we talk about slowing aging, we mean extending health span and not so much lifespan, although they may go together to some extent. And so first I'll tell you a little bit about the program we're doing at National University of Singapore, and then I'll move on to 3 little stories about translation and aging. What we tried to do is to create a full pipeline at the university. We have about 40 faculty now working on aging, and they do everything from basic and preclinical science to translational research to clinical science, and all the way through to educating the public about what longevity means, what longevity medicine means, and why that might be beneficial to think about in this society. Our approach is really, from the translational perspective, is really agnostic. So yeah, I certainly have my favorite molecules that might slow aging, but there are many of them out there, and we want to test as many as possible in a non-biased way. We also want to be very collaborative, working with other academic partners and private sector partners. And so if you have thoughts on how you might want to work with us, we're happy to discuss that. And I think a key thing is that we want to get human. So the part of the aging research field that I come from has really been about trying to understand the mechanisms that drive the aging process. A lot of that's been done in animal models. I'll show you a little bit of animal models today. But I think we've reached a point in this research that we now need to try to determine whether the strategies we have will really alter the aging process in people. So on the preclinical research side, we really have all kinds of different systems, from non-vertebrates like yeast and worms and flies, which have surprisingly been a driving force in understanding the mammalian aging process, to killifish, which are a rapidly aging fish model, stem cell models, organoid models, a lot of work in mice, and a lot of bioinformatics. So we're doing a lot of artificial intelligence, trying to look at big data and understand what aging means from a molecular level. One of the ways we're doing that is to look at systemic biomarkers of biologic age, and I'll talk more about them as we go on, and also to align those biomarkers with individual pathways or hallmarks of aging that have been proposed. We want to really connect the dots between the pathways that drive aging, the systemic measures of aging, and the diseases of aging. On the mouse side, we're realigning how we do research. So instead of giving mice some intervention and waiting until they die, we're starting at 18 months of age, more or less a 60-year-old human, doing about 6 months of treatment, and then looking at frailty in mice as well as molecular biomarkers of age to determine whether we're slowing the aging process. And so this strategy is very aligned to how we're doing our human clinical studies. And the idea there is that if we align these two things, then we can make mice a better model of human outcomes. And we'll let you know how that goes. And then, of course, we want to get human. And so in Singapore, I didn't show any of the statistics today, but basically over 25% of the population is going to be over the age of 65. And when you couple that, and that's in 10 years, and when you couple that with a birth rate among the lowest in the world of 1.1, you have only 2 working people for every retired person. And so that means there's a mandate to really figure out how to keep people healthy and functional longer in Singapore. So we want to take these interventions that have been proposed and validate them in humans. Ultimately, we need to optimize and personalize them, because what works for one person may well not work for someone else, and we need to understand why that's the case. And then we want to scale. And so what that means is that if we're going to scale something, We can't have a $200,000 procedure that only rich people can afford. What we want are cheap interventional strategies to keep people healthy longer and relatively cheap biomarkers where we can measure the rate of aging in the population. Ultimately, we'd like to be doing this for all 5 million Singaporeans. In terms of biomarkers, we're looking at a wide range of different things. The interventions are 6 months long, so either in healthy people or in early pre-disease people. So 45 to 65-year-old population, not the frail population later. These interventions may help them as well, but we think it's the best place to start is the people that are not yet having multiple chronic diseases, but are at risk of getting those diseases in the very near future. And then we're going to look at a number of different kinds of interventions, including diet and supplements, but also repurposed drugs, exercise, and other lifestyle modifications. So we want to do 10 or 15 different interventions and compare their relative outcomes to each other for different biomarkers of aging. So some of the studies we already have going, we have 450 Singaporeans across 3 ethnicities, deeply phenotyped for biologic age, and we're analyzing that data now. We've got a collaboration with the National Precision Medicine team to measure DNA methylation, telomere length, and other aging biomarkers in 10,000 Singaporeans. That data is further along in the analysis, and there's going to be some interesting findings with respect to that. And then, as I said, we're diving into intervention studies, and we can talk about that more. And then finally, we have many different collaborations with other hospital departments and other individuals and companies working on aging. Now, there are no shortage of potential interventions in model organisms that might affect aging. If you look at the drug age database, there are over 1,000 distinct drugs. 110 of those are reported to do something to aging in mice. And these ones on the left here are ones that we work on in my lab. So I'm going to tell you a little bit about alpha-ketoglutarate and gemfibrozil, which are 2 different kinds of small molecules. But I also want to point out that these molecules in green, urolithin A, NSAIDs like ibuprofen, rapamycin, spermidine, vitamin A, and glycine, in our hands, all slow aspects of aging in mice. And so if you look at the different things that have been reported, we can repeat a lot of them, not everything, but a lot of things actually work in our hands. And I think that's a good sign that there's some of these interventions are robust and likely to have a chance of working in humans. So I'm going to talk about AKG just briefly because we have human data on that. And this started with a collaboration with a company that I'm involved with, so that's a disclosure, Ponce de Leon Health. And their idea was to find combinations of natural products that would have additive effects on lifespan and health span. So you take natural products, start interventions at 18 months of age. In this case, we did look at survival in the mice and a number of age-related phenotypes. The product on the market is alpha-ketoglutarate plus low-dose vitamin A in men or low-dose vitamin D in women. But the mouse data I'm going to show you, which was published a couple of years ago, is only on the alpha-ketoglutarate, not the vitamins. And this is the key finding. It's a bit of a complicated slide. The blue dots are control animals. The pink dots are alpha-ketoglutarate treated animals starting at 18 months of age and moving out. And you can see that each dot is an animal. The higher the score, the higher the frailty. And this frailty in the control animals is going up faster than the frailty in the alpha-ketoglutarate treated animals. But this is a complicated way of looking at this data because this dot right here at 23 months is a very frail control animal that died before 25 months. So it's not on the frailty score. So we're losing animals throughout this analysis. So what we did is go back and replot the data as a percentage of the lifespan of the animal where we took the frailty score, 50 to 60, 60 to 70, 70 to 80, etc. And what you can see is the frailty goes up much faster in the control animals than the AKG treated animals. So we see about a 50% reduction in frailty with alpha-ketoglutarate. There's about a 5 to 10% extension in lifespan. So the effect on health span is much bigger than lifespan, and we think we're compressing morbidity. So we went on to human studies. So this is the first one, which we published. And this is not a placebo-controlled study, just to keep that in mind. There are issues with that because of that. But this is users of the Ponce de Leon product. There were 42 people in the study. average age around 60. And the outcome was interesting. So on the left here, we're looking at biologic age, or actually chronologic age minus biologic age. So as the dot lines go up here, that means people are getting biologically younger in the experiment. So what we saw is people taking this product for about 7 months got about 7 years biologically younger using DNA methylation age as a biologic clock. Now, again, I think there is a placebo effect. So what we're looking at here probably is a combination of placebo plus the effect of the product. And so we are going back to try to tease those things out. But I think there's one point I want to make from this study, and that's that there were 2 factors that predicted the response of the individual. One is their chronologic age at baseline. And surprisingly, at least to me, this product worked better in people that had higher chronologic age. We thought that maybe once people got to a certain point, they wouldn't respond anymore because it was too late. But in fact, they responded better. The other people that responded well were people that had a biologic age higher than their chronologic age. So that means that they were not aging particularly well, at least by this DNA methylation clock. And that was dramatically reversed. The people that were already biologically young had a much smaller response to the product. So we went back and did a placebo-controlled study. This is by the company, done at Indiana University. My lab is just analyzing data. We had no part in the collecting of the data. 9-month intervention, 6-month crossover, about 100 subjects, which is a fairly big study for a supplement company, middle age. And we chose people incorrectly for this study because we didn't have the results of this study showing that it was the biologically old people who responded. What we chose are completely disease-free people in Indiana in the middle of a pandemic. And what we found is if you use this clock, which is Morgan Levine's PhenoAge clock, a different clock from the last one, and you look at the people that come into the study that have no disease, they're already about 6 to 7 years younger than their chronologic age. So let me say that a different way. If you're in the US and you're middle-aged, and most people already have chronic diseases by their 50s and 60s. So if you select the group that don't, you're picking the individuals that are biologically very young to enter into the study. And so what we saw is almost exactly what we found in the previous study. Here's the response of people on the Rejuven product. This negative means people are getting biologically younger by the Levine clock. For every 3 months that they're taking the product, they get this much younger. So this person gets 1 year younger for every 3 months on the product. And what we found is for the people that were already biologically young, so these people were already 5 years younger than their chronologic age or more, there was almost no response to the product. But everyone that was within 4 years of their chronologic age or older responded dramatically to the Rejuven product. So this result, in a controlled way, recapitulates what we found in the previous study. And I think we're fairly confident now. We have 3 different clocks that show the same thing, that we're having a positive effect on people if they're around their normal age or not aging well. The people that are already aging very well may not be responding that much. Whether that happens for other interventions is unclear. So I want to move on to one more quick study. And then 2 more really quick studies. The first one's, I wanted to talk about a repurposing drug because the last one was a supplement. So this is another example of molecules that people are trying to test to see if they slow aging. And this one is Gemfibrozil, which is a fibrate I'll talk about in a minute. It extends both lifespan and health span in yeast. That's how we got interested in it for reasons I won't go into. And in worms on the right. It also extends health span in the worms, so it's a conserved effect on longevity. And so that led us to look into this in more detail. So Gemfibrozil this is the drug over here on the left. The trade name is Lopid. It was actually put on the market in 1976 as a hypolipidemic drug. This is the effect here. It lowers triglycerides in the population and raises HDL cholesterol, which is a good thing. And so it's still widely used for management of hyperlipidemia. Now, the mechanism of action, and I should say that this is work by a postdoc in my lab when she was at the Bach, Chong He, and also Shermaine, who's a wonderful graduate student in US. The drug is supposed to get into the cell, disrupt this trimolecular complex, free PPAR alpha, which is a transcription factor, to go to the nucleus and regulate a lot of different transcription events that lead to changes in lipid levels in the cell. So that's the idea, and that's what fibrates as a general class of drugs do. However, what we found is that other fibrates don't affect aging, only Gemfibrozil. And our data suggests that PPAR alpha is not the target of Gemfibrozil. We think it was misclassified, and we've identified other targets now. One clue as to what Gemfibrozil was doing is that it reduces amino acid uptake. And you could do this experiment by asking yeast to grow on all kinds of yeast cells now, we're talking yeast, to grow on different kinds of carbon sources. And what we found is that when you ask the yeast to go on dipeptides and tripeptides, Gemfibrozil blocked the growth of those yeast cells, but they grew fine on other kinds of carbon sources. And that suggested that these dipeptides and tripeptides are not getting transported into the cells. And indeed, if we look at amino acid levels inside the cell, almost all of them are reduced in the presence of Gemfibrozil. And finally, yeast are cool because you can do all kinds of elegant experiments. And here's one. You take a toxic dipeptide that will kill yeast if it gets into the cell. You put it on this piece of paper and let it diffuse into the media, and the yeast that are around it all get killed by the toxic dipeptide. If you use Gemfibrozil, though, you block that uptake, and that protects the yeast cells, and they can still grow. So we're fairly confident that's what's happening in yeast. We see the same thing in worms, where you block amino acid uptake with the use of Gemfibrozil. And in human cell culture, we find similar things where you can reduce the levels of amino acids in the cells by adding the drug. And so that's one mechanism we think is affecting things, and we can even extend that to the intestinal experiments in mice. So we take out the intestine, incubate it with fluorescent dipeptides in the presence or absence of the drug, and then look at the effect of this. And what we find is that over here, the vehicle, you still get a lot of dipeptide uptake. You can block it, though, with losartan, which is a known positive effect. But you can also get that with Gemfibrozil. So this is a very robust, I think, set of data at this point. And if you're going to reduce amino acid uptake into the cells, the downstream effect of that is you'll reduce mTORC1 activation. This mTORC1 complex is the most conserved complex that linked to longevity. Reducing TOR signaling extends lifespan almost everywhere it's been tested. And so we could show that here. This is a complicated slide, but if you starve cells and look at the TOR kinase, looking at S6 kinase phosphorylation, it's very low, not affected by Gemfibrozil. You add amino acids, you activate TOR, it's not affected by Gemfibrozil. But if you add dipeptides, you can activate TOR, but now you block the effect with Gemfibrozil. So we think that this drug is reducing mTOR signaling. So let me just add that we have 3 different targets that we think are the real targets for this drug. And the next step now is to modify the drug to find new compounds that hit the real targets better, either for extending health span and targeting aging directly, or for treating diseases like hyperlipidemia. And so we'll be doing that. So I want to just tell one more story to take a step back, because it sounds like we know everything about aging, but in fact we don't. And in reality, we've only explored a very small segment of the aging pathways, I believe, and the ways to slow aging. There may be many, many that we have not discovered yet. So we wanted to take a step back, and Matt Kaeberlein and Jan Gruber and I formed a company to do high-level drug screening using worms, C. elegans. We have 2 wonderful postdocs from Matt's lab. We're working on this, really 3, Mitchell, Ben, and Jason. So we'll just tell you what we're doing here to give you a feel for this. And what Matt's done is created this machine called a WormBot, which does high throughput, unbiased longevity drug discovery. So in other words, we can screen thousands of worm different conditions on one robot in a very short period of time. The idea now is to make an army of these WormBots, where we'll have 50 of them. And if we can do that, we can screen hundreds of thousands of drugs every month. the point is to find new drugs that haven't been known to affect longevity before, but also to find combinations of drugs that work together to better affect longevity. And so I'll give you one example of that. This would be what 40 wormbots could do, 100,000 experiments a year. Each experiment's a screen. And just as proof in principle, we took one of these, took the FDA-approved drug library and took a disease model in worms called the amyloid beta model. So this is a toxic peptide that is linked to Alzheimer's disease. You probably know of. It forms aggregates in worms and causes rapid paralysis. So what we did is, rather than just screen the drugs alone, we screened them either alone or with metformin, which has a modest positive effect on its own, to look for combinations. And I'll show you one of these cool combinations. I have this thing here. So what we're looking at here is the effect of metformin in red, the combined effect of the FDA-approved drug in blue. So some of them are beneficial, and then the combination in green. So this is a good example here where you have sorry, let me find it here. You have a small effect of metformin, no effect of the drug alone, and you combine it, you have a dramatic effect. So let me show you what that looks like. So here we have, if this plays, the video is probably not going to play for some reason. Yeah, I think I'll just skip this slide for the sake of time. But these worms with the drug and the new drug and metformin can stay active for much, much longer than either alone. And you can see this kind of synergy here. This is in terms of looking at the effect of the drugs. 3 different drugs combined with metformin have a synergistic effect. So we think there's value in finding combinations of small molecules, and we're looking hard to find these. And then finally, I just want to close by saying we also have a Center for Reproductive Longevity in the US, which is heavily interested in ovarian aging. And I'm happy to talk to anybody about some of the projects we're doing there offline. Finally, we have a webinar every Thursday at 7 Singapore time, but all of the shows are available on YouTube for free afterwards. We have about 65 of them now. We have about 500 attendees for each show, and then we have up to 100,000 viewers on YouTube for each show afterwards. So what we're really trying to do here with this webinar is bring together scientists, clinicians, interested parties, investors to really move forward this longevity medicine approach. The website's down here. If you need, you can email me and I'll send it to you. And with that, I have a lot of people working in the lab. I have a great collaborator in Andrea Meyer at NUS, who's a clinical expert in gerontology. We're working together to try to tackle this aging problem. And I just want to thank you for your time. Happy to take questions.